So now we are going to discuss the physiological or biochemical test. The purpose of these tests, of which there are many, is to determine the enzymes that are present or absent in a particular bacteria cell. This allows us to gain important information about each bacteria cell and their characteristics. These tests also provide information on the metabolic characteristics of a cell. So in this unit, we've been talking about metabolism. Metabolism is a fancy way to say, can a bacteria take a particular food source, whether that's a sugar, a protein, a lipid, etc., and convert that food source into energy or ATP? By doing physiological or biochemical tests, it allows us to get a look into what's happening enzymatically inside that bacteria cell. It allows us to see what kind of metabolism is occurring and what kind of properties that bacteria allows. These tests also provide us with information on the foods or substrates a bacterium likes to eat or utilize. The purpose of these tests is to gain information about the bacteria. It also is going to aid in our ability to identify an unknown bacteria. For example, if there's a food outbreak that's bacterial based, we can do certain physiological tests, and based on the outcome, we can identify and then treat our patients that are part of that outbreak. So throughout these PowerPoint slides, we are going to look at the different physiological tests that we are going to study this semester in this course. There are many other tests that we are not going to look at that also play an important role in the metabolism of certain bacteria. It is important to note that not all bacteria possess the same enzymes. So we're gaining information on seeing what enzymes and metabolic processes are occurring within the cell. So now we see a figure or diagram that looks very daunting. This figure is also located in the lab manual. What this is an overview of how all the pieces and the physiological tests that we will be testing fit together. This is going to show a metabolic chart or the physiological or biochemical test that we will study in lab. What this really means is the metabolism of an individual microbe, specifically bacteria. All microbes need to make ATP, and they do it slightly different depending on whether they are anaerobic, aerobic, or do fermentation. We will study in the lecture portion specifically aerobic metabolism. We will also look at anaerobic and fermentation. All three of those types of metabolism start with glucose, located here on our picture. So glucose is a monosaccharide. One of the tests that you will be looking at is the phenol red or PR test for sugars. That sugar will be glucose. If glucose is fermented by a particular microbe, then it will be broken down into an acid called pyruvic acid, and a color change will be seen from red to yellow because of the indicator tie phenol red. Also, if fermentation has completely occurred, gas will be produced, and that will be seen in the little inverted Durham tube. Another test that we will be doing is a starch hydrolysis test. Many bacteria possess the enzyme amylase to break starch down into individual glucose molecules. Here you can see how those two pieces fit together. For this particular semester, we will not be studying lactose, which is a disaccharide. But lactose, which we'll study later in this course, can be broken down by certain bacteria by an enzyme called beta-galactosidase and then used for glucose for aerobic respiration. Another test we will look at is the citric acid or Simmons citrate test. The citrate test looks to see if a microbe has the ability to use citrate as its sole carbon source to produce certain products, and the pH will go up. Citric acid, as you can see, is part of the Krebs cycle, which is also part of aerobic respiration. Two other ones to mention, we will be doing something called the mixed acid or methyl red test, in which we can look to see if acids are produced. We will also do the Voges-Proskauer or butanediol test to see if this particular alcohol is produced. Last but not least, we will look at a test called the nitrate reduction test, which has to do with whether or not a bacterium possesses the enzyme nitrate reductase. This is just a brief overview of how all of these physiological tests play a role in metabolism, which is crucial to the survival of a particular bacterial cell. So the first test we're going to discuss is called the catalase test. This test is used to differentiate or put into two different categories, Staphylococcus versus Streptococcus. Both of these groups of bacteria are gram-positive cocci. However, one possesses the catalase enzyme and the other group does not. So what is catalase? It is an enzyme. Enzymes often end in ACE or ASE. The role of many enzymes is to catalyze a chemical reaction. In this case, the enzyme catalase job is to break down H2O2, which is hydrogen peroxide, into water and oxygen. In many bacteria, hydrogen peroxide is toxic and would cause disruption in proteins, DNA, and RNA. So if a bacteria possesses the catalase enzyme, the enzyme breaks H2O2, which is hydrogen peroxide, down into water and oxygen, which are now harmless. So it is an advantage of a bacteria to possess this enzyme. So for each of these tests, we have a certain set of criteria. And in your lab information, you will have a table. This video will help you fill in that table. So listen closely and you will be able to fill it in based on our discussion. So the first category is culture medium. So typically that means what do we add the microbe to? But for this test, we actually don't need to grow any more bacteria. This test we can do immediately if we have already a test tube of bacteria or a plate of bacteria. So we don't need to have the culture media, the initial appearance, or how to inoculate. Those steps are not necessary for this particular test. So what you do need for this test to occur is you need a test tube. Triptychsoid agar or nutrient agar are typically the two used, with a bacterium growing on the surface of the slant. Once you have the bacteria, you will need a few supplies on the next slide. Catalase test procedures and results. The test procedures for the catalase test includes adding chemical reagents to see if we have positive or negative results. So what you would do is you would take your initial test tube of bacteria. Again, there's no incubation time if you're given the bacteria. You would take your test tube of bacteria and you would label one or potentially two clean glass microscope slides. You would label them with the name of the bacteria. Then aseptically, using your loupe, you would add a small amount of bacteria to the correctly labeled portion or microscope slide. Next, add 1 to 2 drops of hydrogen peroxide to each bacteria and view immediately for the presence or absence of bubbles. The chemical reaction for the catalase test is very important. We have 2 H2O2, in other words, 2 molecules of hydrogen peroxide, potentially being broken down into 2 water molecules and oxygen. The oxygen would be seen by the presence of bubbles. This would indicate that the enzyme catalase is present. So once you've added that hydrogen peroxide, if you see bubbles, which is the positive result, that would tell us that we have oxygen being released and that catalase is present and this reaction did occur. The negative result, which is why it has an X over it, means this reaction did not occur and that catalase is absent. A negative result would be seen or indicated by no bubbles. The bubbles occur almost immediately, so it is important to take any kind of documentation such as photographs or notation right after you've added the hydrogen peroxide to see if it's a positive or negative result. Next, we will discuss the nitrate test. This slide is going to show what the broth looks like before inoculation. The culture medium and the nitrate test is nitrate broth. Its initial appearance is clear or a very light pale yellow broth. As you can see by the picture on the right, it is uninoculated, which can be indicated by the lack of turbidity. You're going to use an inoculating loop to inoculate one loop of bacteria into the broth. This slide shows what happens after 48 hours of incubation. The test procedures for the nitrate broth are to add the reagents nitrate A and nitrate B. It should be noted that these are very toxic chemicals, so this should be done in the hood wearing safety glasses and gloves. So to your 48-hour incubated nitrate broth, you're going to add 2 to 3 drops of nitrate A solution and 2 to 3 drops of nitrate B solution. Then very gently shake the tube to uniformly distribute all of the solutions so they can get mixed up. The chemical reaction for the nitrate test is to reduce nitrate to nitrite. If this can occur, then the enzyme nitrate reductase is present. You can see the chemical formula, which is nitrate, which is NO3, is reduced to nitrite, NO2, by that enzyme nitrate reductase. A positive result will be seen if the entire test tube is red. This indicates that nitrate has been reduced to nitrite and the enzyme nitrate reductase is present in that particular bacterium. A negative result will be seen if the solution is a cream color with little red dots, sometimes called precipitates. No matter how much you shake the tube, if it's a negative result, the entire test tube will not turn red. So with little tiny dots of red, this is the negative result. The entire test tube must turn red and remain red for the positive result. For the negative test, nitrate is not reduced to nitrite, and therefore the enzyme nitrate reductase is absent in that particular bacterium. Let's begin by talking about starch. Starch is a polysaccharide or many sugars that can be broken down by amylase to produce the monomers or glucose. If you were to take any product that has lots of starch, such as a starch plate or a potato, and add iodine, the iodine will react with the presence of starch, turning a dark brown color, indicating starch is present. In this experiment, we are actually going to look to see if the bacteria possess the enzyme amylase and look for the absence of starch. In other words, if there's an absence of starch, we will see where the iodine did not react, which will appear as this halo. The absence of starch indicates the presence of the enzyme amylase. So you will obtain a starch plate, which is a plate of starch agar. For the initial appearance, it has no growth. The inoculation is very simple, which is to add one loop or one streak of bacteria down the middle of the starch plate. You will then incubate the plate and add iodine after 48 hours of incubation. After incubation, the bacterium has had time to grow. It should be noted that you might see growth on a starch positive or starch negative plate. What we're going to look at is the presence of amylase, which cannot be detected until iodine has been added to the plate. So after 48 hours of incubation, you'll take your plate out of the incubator and you're going to add enough iodine to flood the plate with the iodine. The chemical reaction is starch is broken down into, which is the arrow or yields symbol, into glucose. Starch is the polysaccharide and glucose is the monomer. This occurs by using the enzyme amylase. As you can see by the picture letter A on the left, if amylase is present, a halo will be seen around the growth. The halo indicates the iodine is not reacting with the starch because the starch is absent. The starch is absent because the bacteria contained amylase and was able to eat the sugar, in this case starch, and therefore the starch is gone. In letter B, the negative result, there is no halo because starch is still present and amylase is absent. So our next test actually kind of goes back to our triple sugar iron. It's just slightly different. So we're looking to see if bacteria are able to eat or ferment different types of sugars. In these test tubes, we have something called phenol red. Phenol red is an indicator or dye that's going to tell us whether or not that sugar was fermented or eaten. So as we can see, many bacteria can ferment or metabolize various sugars. So in this experiment, we could use glucose, which is a monosaccharide. We could use sucrose, which is a disaccharide, or lactose, which is a disaccharide. We could use any of these sugars. And we could inoculate these cultures, which are all going to start off as a red color because phenol red is our indicator. They're all going to start off red. And we would inoculate by adding 1 loop of bacteria to these test tubes. If an acid is present, we will see a color change from red to yellow. If gas is present, we will see a change in this little Durham tube, which I'll get to in a second. Those two indicators, acid and gas, tell us whether or not the bacteria are able to ferment or metabolize various sugars. A Durham tube is this little inverted test tube. So here we have our little inverted test tube. And initially, it's going to be full of media. So in this case, full of, let's say, glucose broth. And again, we kind of have that red appearance. The Durham tube is this inverted test tube full of media. So our culture tube is a phenol red sugar broth. That sugar could vary. It could be glucose, lactose, or sucrose, depending on what's available. The initial appearance is red, as you can see. And that we need to know the color, and we need to know whether or not this Durham tube is full. And this one's all full, indicating it's ready to go, ready to inoculate it. How do we inoculate? We're going to add 1 loop of bacteria using our loop aseptically to our test tube. So 1 loop of bacteria to our test tube. And again, you should record this data on the corresponding worksheet. So now let's look at the results of our phenol red sugar fermentation tube. It should be noted that we could use any sugar, glucose, lactose, sucrose, etc. We chose to use glucose for this particular example, but the actual sugar does not matter. So we inoculated our tubes initially, and now incubation has occurred. It is 24 to 48 hours later. So for this test, it's a visual test. All you need to do is look at the appearance of the tubes to determine positive or negative results. However, there are 2 components to this test. The first is going to be whether or not acid has been produced. So we're going to look for the color, and if acid has been produced, we would see a yellow color. The second part of this test is to see if gas has been produced. If gas has been produced, the Durham tube, which is the inverted test tube on the inside, will not be full. So let's look at our options. So here we have our phenol red sugar tube. Phenol red is the indicator dye within the tube that's going to tell us whether or not acid has been produced. It's a sugar tube because it could have glucose, lactose, sucrose, etc. One of those sugars within these tubes. So the first one we're going to look at is for acid production. So this first tube appears red. So there's red. And the Durham tube is also red. Now, this one looks a little kind of orangey. It's just from the photograph. But as long as the majority of the tube is red, that is going to be negative for acid. The second part is we're going to look for gas production. So that Durham tube is that inverted test tube that initially was full of media. It is still full of media, and therefore we are going to be negative for gas, specifically negative for carbon dioxide production. So let's talk about the explanation of these results. If it's negative, negative, that means the sugar was not fermented, and therefore that particular bacteria did not have the enzymes to metabolize this particular sugar. So let's go to the second option. We have another phenol red test tube, and this test tube appears yellow. So if the test tube is yellow, that is going to be positive for acid. If acid is present, that phenol red indicator is going to change from red to yellow. So I see yellow, so positive for acid. However, the second part of this is for our gas production, and that Durham tube appears to be full. So that would be negative for gas. So that's a second combination possibility. So again, positive for acid because of the yellow, negative for gas because that Durham tube is full. So what does that mean about our explanation? What that means is the bacteria was able to partially ferment, metabolize that sugar. So it was able to do enough to produce some acid, but not enough to make any gas. So our third option is we see, again, our yellow color. So that phenol red is telling us we're positive for acid because we see yellow. And then our Durham tube, we also see it's being partially empty. If there's even a small bubble or space, that is going to be a positive result. So never take a tube that has a bubble in it to start with before you inoculate, because that would be a contaminated tube. So any bubble, large, small, etc., indicates that gas has been produced. And so I'm going to highlight that a little bit more. You can see right here with this blue arrow that the Durham tube, again, is that inverted test tube, is not full. So if we are positive for acid and positive for gas, what does that mean about the bacteria? Our explanation. That means the bacteria possess the enzymes to fully ferment the sugar. And by fermenting or metabolizing that sugar, it was able to produce acid and gas. So our next test is called the citrate test. This test is commonly used to differentiate different types of gram-negative bacteria. In this test, we will be using citrate as the sole carbon source and determining whether or not the bacteria can use citrate and ammonium phosphate as the nitrogen source. So you should recall that the citric acid cycle is part of metabolism, glycolysis, our prep step, our transition step, and then we get to step 3, our citric acid cycle, our Krebs cycle. So this test is going to basically tell us a little bit more about that cycle. Initially, in this test, you will start off with a test tube of something called Simmons citrate, and it has a green color and a pH of 6.9, which is basically neutral. So 6.9 is almost 7, and 7 is neutral. That is the color initially. Then, if the bacteria is able to use citrate as the sole carbon source, and ammonium phosphate, ammonium again is NH3, that would cause the pH to go up, making it more alkaline to about our pH of 7.6. That would indicate a blue color by our indicator called bromothymol blue. We will look at this more on the next slide. So our culture medium, as you fill in the corresponding worksheet, is Simmons citrate agar slant. The initial appearance is a green agar slant. How to inoculate? We would use 1 loop, again aseptically, to add bacteria to the surface of the slant. So right with our loop on the surface of the slant. Then we would get more bacteria aseptically, and we would stab all the way to the bottom, the butt of the slant, similar as we did in the triptych sugar or triple sugar iron. We would go all the way to the bottom, and we would repeat this process twice, one using the loop and one using a needle. So now let's look at our test results. So the test procedure is not going to require any chemical agents. We would look at the color of the test tubes or look at the tubes. And here is our chemical reaction, which is very important. Chemical reaction right here. We have citrate in our tube. We have ammonium phosphate in our tube. We're looking to see if ammonia, which is going to increase the pH by that bromothymol blue indicator, has occurred. So here was our initial test tube, which is green, with our pH of about 6.9. So we have fairly neutral pH in our first test tube. Then if we have a positive result, which we can see here, so here's our negative result. Here's our positive result, which is our blue color. The blue tells us, because of the indicator of bromothymol blue, the pH went up to about 7.6, which is more alkaline or basic, moving that pH up. So a positive result tells us it's a blue slant and that the bacteria was able to use citrate as its sole carbon source, and it was able to make ammonia, causing that pH to go up. Our positive result is this pretty blue color. A negative result looks like it did before inoculation. In other words, it's an unchanged or it's not a different color. So in this case, our bacteria did not utilize citrate. Ammonia was not produced. The pH remained 6.9. Our last two tests go together in that they both use the exact same culture medium, which has the same appearance, and you inoculate the same way. So let's look at each of these tests. The first one is called the methyl red test. It is also sometimes called the mixed acid test because we're looking to see if acid is produced. Before inoculation, the culture medium is MRVP broth. The MR stands for methyl red. It is a clear, dark yellow broth, and you inoculate using one loop of bacteria. After 48 hours, it is important to note that you would need to separate the broth into two different test tubes. The two tests have different chemical reagents, and if you only add the reagents to the initial or original test tube, you will only be able to perform one of the two tests. So after you have inoculated the MRVP broth and they have been incubated for 48 hours and allowed to grow, the next step would be to pipette 1 milliliter of the original MRVP broth into a new clean test tube labeled VP. The original test tube, after the pipetting has occurred, will now be used for your methyl red test. The new clean test tube labeled VP will be used for the VP test. The methyl red test, after incubation and after the separation of the 1 milliliter into a clean test tube, we will now use the original MRVP broth to perform the methyl red test. To the methyl red or MRVP broth, we're going to add 2 to 3 drops of methyl red reagent. Then shake gently. The chemical reaction is glucose, which is what's in that MRVP broth, could be broken down or fermented into pyruvic acid. If this occurs, then acidic products will be seen. Think about our pyruvic acid that we got from glucose in aerobic respiration. If more acidic products are produced, like lactic acid, acetic acid, formic acid, then when we add the indicator dye methyl red, this will turn red, indicating acids have been produced. A positive result for the methyl red test is glucose, which was in the MRVP broth, being fermented to pyruvic acid and producing more acids. This will produce, as we can see over here on the right, a red result. This one's fairly easy to remember. Methyl red uses methyl red reagent, and a positive result is red. A negative result will almost look identical to the initial broth, which is that pale yellow color. A negative result indicates acidic products were not produced. And instead, when we add the methyl red, it will turn a little bit more yellow, indicating a negative result. The VP, or Vogts-Proskauer test, sometimes called the butanediol test because we're looking for butanediol, has the same culture medium as the methyl red. So it's MRVP broth, which is still a pale or dark yellow appearance, and we still inoculate using 1 loop of bacteria. After incubation for 48 hours and after we have separated the broth into the 1 ml test tube, the new clean test tube, that little 1 ml will be used for the Vogts-Proskauer or butanediol test. This test takes a little bit of time, so sometimes it is recommended to perform it first. You're going to add 18 drops of something called Barrett's A and 18 drops of Barrett's B. Shake gently until it gets nice and bubbly or frothy. Your lab instructor will show you or demonstrate how to shake the tubes safely. All of these reagents should be added in the hood with gloves and goggles on. The most important part of this test is to let it sit for 15 to 20 minutes. The longer it sits, the more it will react. If you do not wait, you will see a false negative result. In the Vogts-Proskauer test, the chemical reaction is glucose, which is still our MRVP broth, is going to be fermented or broken down into pyruvic acid. But now, instead of producing more acids, as we saw in the methyl red test, instead if the bacteria is able to produce alcohols, such as butanediol, a neutral pH will occur. A positive result would be glucose broken down into pyruvic acid and producing butanediol, an alcohol. This will give us a deep red, like a wine red color, indicated here on the far right picture. And again, the longer it sits, the darker the color is going to be. That shaking is also very important. So this one takes about 30 minutes after we've added the reagents. A negative test turns kind of a green-brown color, seen here, and glucose is broken into pyruvic acid, but we do not produce butanediol or neutral pH. It should be noted that a single bacteria cannot produce an acid and a neutral pH. What that means is a single bacteria cannot be positive for methyl red and a positive for the Vogts-Proskauer test. They will have opposite results.